Good morning. Today we are group A4. We're doing bioremediation techniques and uses.
My name is Saida Gomez, and I will be doing what is bioremediation.
My name is Sienna Jones. I will be doing the types of bioremediation.
My name is Adrian Lee Vasquez. I will be doing techniques.
My name is Melanie. I will be doing bioaugmentation and the type of techniques we can use for different types of waste.
My name is Eli Fazell, and I'll be doing the future directions and conclusion.
Bioremediation is when an organism such as prokaryotes, fungi, or plants are used to restore a contaminated or damaged environment. The environment can either be soil or water. Bioremediation is important because it cleans up the environment from toxic pollutants. This results into having more richer and healthier waters, preventing further contamination.
One type of bioremediation is microbial bioremediation. It is the use of bacteria, fungi, or archaea to break down or neutralize pollutants in the environment. Microbes can metabolize harmful substances, including oil spills, heavy metals, pesticides, or other toxic wastes, and convert them into less harmful or non-toxic byproducts. There are multiple techniques to microbial bioremediation, including bioventing and biosparging. Another type of bioremediation is intrinsic bioremediation. Intrinsic bioremediation is a process where indigenous microorganisms in the environment degrade or neutralize contaminants without human intervention. It relies on the native microbial communities that have adapted to break down pollutants over time, using them as energy or nutrient sources. The difference between intrinsic and microbial bioremediation is that intrinsic bioremediation happens naturally, while microbial bioremediation requires people to introduce the microbe into the environment. An example of intrinsic bioremediation is phytoremediation.
Bioventing is a natural and cost-effective way to clean up petroleum-contaminated soil. By injecting air into the ground through the vasozone, we provide oxygen that stimulates microbes to break down petroleum hydrocarbons. This process enhances natural degradation without the need for harsh chemicals, making it an eco-friendly alternative to traditional treatments. It's a simple yet effective solution for restoring contaminated sites while minimizing environmental impact. Phytoremediation is a green, cost-effective way to clean up contaminated soil and water using plants. These plants absorb, break down, or stabilize pollutants like heavy metals and hydrocarbons, naturally restoring the environment. It's an eco-friendly alternative as well to chemical treatments, reducing pollution while also improving soil health. By harnessing the power of nature, phytoremediation provides a sustainable solution for long-term environmental cleanup, although it is a slower process compared to other types of techniques.
Some limitations and challenges are how bioremediation can be time consuming as the organism takes long periods of time to clean up an area. This results into the decontaminations to happen in expanded times. Another limitation could be how there are specific types of contaminations that would require a certain type of organism. Because of this, it would be difficult in choosing the most effective for the pollutant.
Bioaugmentation. Bioaugmentation has been around for thousands of years at this point. In ancient Greece and Rome, they used to direct wastewater through complex networks of channels and reservoirs to create an environment ideal for microbes to grow in there. And in the process, they would break down organic matter and organic contaminants that were in that wastewater. Microbes consume contaminants and excrete less harmful compounds. And whatever the contaminants that are released from those microbes, those get removed, and they're also more cost efficient for companies to do. Bioremediation applications for soil and water. In soil, we can use nitrifying bacteria, which break down the original compounds into nitrates. Those are harmful for plants, so we need to use bacteria like nitrobacters to turn the nitrate into nitrites into nitrates. Those are beneficial for plants, and therefore we can release them back into the soil. After the Hiroshima, Fukushima, and Chernobyl nuclear disasters, sunflower fields served to clean up the soil. There were thousands of acres of sunflowers that were grown just to remove the some of the more harmful elements that were in that environment, which was cesium and strontium. In contaminated water, it is very harmful for humans. It is harmful for plants and animals as well. They are carcinogenic, so it is very beneficial for us to treat the water. We can use bacteria, fungi, algae, phytoremediation, biofilm, and activated sludge. I would like to talk about activated sludge. It is commonly known as a mixed liquor. It is where wastewater and the microorganisms that are going to be used to break it down, they're mixed in an aeration tank because the microorganisms need a very like almost perfect environment for them to thrive. They need a lot of oxygen. Without it, they wouldn't be able to like decompose wastewater as efficiently. And at the end, we end up with a sludge and the separated treated water. The treated water can now be released back into the environment and not be harmful for anyone. In the bioremediation applications used for waste treatment, we consider waste to be garbage, food waste, sewage, water, and air pollutants. Microorganisms usually use a oxidoreduction reaction, whereas where microorganisms acquire energy through catalyzing, they break down the chemical bonds and remove electrons from them. That would be the oxidoreduction reaction. We know that over 90 microorganisms are able to break down plastic. These are 1,000 times more efficient than if the plastic was to just be out in the environment. Because during during my investigation, a single water bottle will take 450 years to break down if it was just out in the soil. Using microorganisms, we can do it in 3 months. So it's a lot more beneficial. I wanted to talk about comamonas because they chew down plastic, and in the process of them chewing it down, they create nanoplastics. They then excrete hydrolase enzyme to break it down into smaller molecules, which they can now consume. And from that, we get things like carbon dioxide, water, and fresh biomass, which then are actually beneficial for their environment.
Eli, you're muted. You're muted.
Got that. Thank you.
Yep.
So, the future directions that bioremediation is heading in is the genomics, transcriptomics, nanotechnology, and gene editing. And the the genomic tool analyze the isotope distribution, the DNA hybrid, hybridization, molecular connectivity, metabolic footprinting, and metabolic engineering. This can help analyze the soil microbial communities and be used to determine the abundance and appearance of taxonomic and operational gene. The transcriptomics, this helps control gene expression, which is critical in adapting changes in the environment that helps lead to survival. With transcriptomics tools, almost every gene can be examined and studied. Nanotechnology has been starting to be used to help purify water. PMUA Nanoparticles are designed to help the water maintain their properties in the presence of diverse range of bacterial populations. Gene editing, the technical developments that allow rational, genetically created fragments at the genome level. This provides the exact spot for addition, deletion, and substitution in DNA molecules. Pollutant-tolerant bacteria are the greatest choice for genetic manipulation. And then for the conclusion, bioremediation is the process of restoring damaged environments by using organism. This cleans up our environment using natural processes, which can lead to richer soil and stop further contamination. And then microbial biomediation is the use of bacteria, fungi, and or archaea cleans up harsh chemicals. The intrinsic bioremediation is a natural process. Native microbes break down and or neutralize contaminants. Bioventing stimulates microbial degradation of petroleum and hydrocarbons, injects atmospheric air into the soil through the phytozone, and then phytomediation is they degrade, stabilize. Oh, give me 1 second. My notes are going all over the place. They degrade, stabilize, or store pollutants in roots, shoots, or leaves. Minimal environmental disruption compared to excavation methods. And then the recap of today's presentations. I did the future directions and the conclusion. Zahid did the what is the bioremediation. Sienna did the types of bioremediation. Techniques was done by Adrian and Melanie. The challenges and limitations were done by Zahid.